So good afternoon to everyone. Thank you for attending. I mean, it's not evident that people attend seminar at 4 p.m. And thank you also, Chris, to accepting to give your talk to our biodiversity seminar. So I'm pleased to introduce Christopher Swan, professor at the University of Maryland in Baltimore. Actually, we don't know each other in person, but I'm following his publication on urban biodiversity and assemblage rule and drivers for several years. And Christopher is more than just an urban ecologist. I would say that he combines research from three main topics, at least from what I got from his webpage: biodiversity and community ecology, ecology of river and streams, and the topic of today, urban ecology. In this context, Christopher will present a paper titled Local and Regional Constraints in Urban Biodiversity, which definitively combine urban ecology and biodiversity and community ecology. So thanks, Chris, again for accepting to give this talk. And I mean, afterwards, we will have plenty of time to interact with him. The floor is yours. Thanks.
Thank you for the kind invitation. It's my pleasure. It's my pleasure to talk about my work. I'm humbled to be able to present today. I'm a community ecologist. It was a very accurate description of what it is that I focus on and most interested in. And today, if I can convince you of anything, it's going to be the role of basically beta diversity in understanding how biodiversity unfolds in the urban environment. And I'm gonna talk about it from in two contexts. One is from a survey point of view, and testing a conceptual model of how biodiversity is organized in urban places. And the other is from an experimental point of view, looking at local and regional effects and how they unfold in the urban context. Whoop. So if you look at urban ecosystems, they're terribly heterogeneous. They are a juxtaposition of habitat patches. And if you take a look across them, historically, very early on, ecologists thought of them mostly as, a bunch of degraded habitats or very disturbed, but we know very well that that is simply not the case. They include lots of built places, lots of managed places, and lots of what I like to call facilitated assemblages, like this upper left image of a basically a landscaped or garden situation where there's plenty of biodiversity here. There's plenty of ecosystem functioning going on, but this assemblage is completely governed by humans. Ecological assembly rules that we use or employ or understand and try to develop in a natural or non-built or non-urban place does not work here. One thing that we need to keep in mind when we understand ecology is the role of dispersal. So ecologists historically, up until about 2004, there was a nice paper by Matthew Leibold where ecologists thought about ecological communities as isolated entities, but they are these ecological communities are subsets of the regional species pool. Of all the species that can arrive at a location, the species that you see are a subset of those species that could arrive from different locations. So dispersal plays an important role. And if you take a look at species richness at a location, this is work that was described by Cadotte in 2006, and look at dispersal rate, we can see that local diversity rises as dispersal increases, as more species show up, but then it slowly declines because you get a dominant competitor or generalist taking over all locations, and therefore regional species diversity also declines. You get competition taking over regionally. And that's why beta diversity or turnover from one location to the next also declines. And remember that according to Whitaker back in 1966 or 67, I think it was, developed the idea that local diversity, alpha diversity, multiplied by beta diversity, which is turnover or species replacement from one location to the next, local times or alpha times beta is equal to regional diversity. I like to think about it like this, is if you take a look at regional diversity as, say, each of these shapes here as being a different species, they unfold along a gradient from dark to light, an ecological gradient from going from dark to light, where each square or rectangle is a different local community. Species sort themselves along this gradient, where you have different species occurring, where turnover is the shift in species composition, and local biodiversity is what you see at each location, and turnover is interhabitat loss and gain. So local biodiversity is what occurs at a specific habitat. Beta diversity is the shift along this environmental gradient. I'd like you to remember this image because I'm going to return to it in the second half of my talk, because this encapsulates local effects, which is the sorting along an environmental gradient, and regional effects, which is this delivery of species from a regional species pool to this effect. I'm going to talk about that in the context of urban places. And I'm going to suggest or offer that ecological theory can't explain biodiversity urban places completely, because people move species around. They're agents of dispersal. They change dispersal pathways. They carry them. They pick them up and plant them. They relax or enhance trophic interactions. For example, they might go to a garden and spray a pesticide and remove herbivores, for example. They sidestep natural patterns of birth and death, weeding, for example, or planting species. I mean, this changes. If you were to create a life table of a species in your garden, you wouldn't be able to do it because there are just no natural patterns of birth and death. And of course, humans change habitat substantially. So the goal for urban ecologists is to better understand the mechanisms generating and sustaining biodiversity in the built environment. And so, and I want to highlight mechanisms here, which is hard. Lots and lots of work right now has described patterns, and there are some conceptual models, and there's some good ones. And colleagues and me and my colleagues have come up with a few. There's some other ones floating around there, and they're good. But I don't think that many have come up with good mechanisms, or at least tested the mechanisms. So today I'm going to talk about two studies. One is to look at woody plant meta-community structure, and the second is to look at local and regional constraints on the diversity of an experimental urban meta-community, and that's going to be zooplankton in stormwater detention basins, which is an odd habitat to think about, but they actually are pervasive around where I live in the mid-Atlantic US. So let's talk about woody plant meta-community structure first. So the conceptual model works like this. I just described to you, this is just a different way of describing it, that the regional species pool gamma delivers species into what I like to call a self-assembled local community, okay? Where you get from one location to the next species turnover, beta diversity. So alpha diversity is a subset of gamma, with beta diversity describing change in species composition from one location to the next. Well, if you place this into an urban context, things become a little bit more complicated, where you get, I'm trying to move my, you can separate the ecological template here in the bottom from a socioeconomic template up top, where strong environmental filtering is imposed by the socioeconomic template, where now we can generate or impose an investment of socioeconomics and policy on community composition, where it might be very low at one end and you've got good ecology going on, where you might have strong environmental filtering by the urban environment, by a strong geophysical template where you might have disturbed or ignored habitat. Moving across here, you might have remnant or conserved habitat. And then where you have very strong investment in socioeconomic, a strong investment of socioeconomics or policy, you get urban design. And here you get dispersal and environmental facilitation, creating strong beta diversity or lots of decision making and lots of variability from one location to the next, giving you what I like to call facilitated assembly of local community structure. And the example is landscaped locations, parks, places where people are imposing very, very strong influence on species composition. And because of the variability in decisions, that translates into variability in species composition. So the difference is that you might have the same number of species, okay, but you have a lot of species in the regional species pool, but the difference is beta diversity is quite strong from one location to the next. And that's the key to understanding why you might have very high biodiversity in urban places. Now, this is a hypothesis, okay? Can we test that? So, using a data set that exists on tree biodiversity in my region, and I'll talk about that in a minute, I focused on two dimensions of biodiversity: taxonomic, or variation in the number of species, and functional, or the range and values for an assemblage of traits that influence ecosystem processes. So this was of interest. Functional was of interest, and taxonomic, of course, makes sense to study here. So what are the predictions? The predictions were that beta diversity would be high where people have a large control over composition, like residential areas, and that we would have a high number of unique species in residential areas and in open spaces. So it would be required that we'd have lots of different locations, lots of information on trees, and in those locations, a lot of variability in land use. And so in our data set, we had different land uses arranged. We hypothesized a gradient in human investment. These land uses were identified based on a known and accepted land use classification. They went from vacant lot to open space, park, institutional, commercial, and residential. And these are the definitions. So vacant lot was where no building had been there since for more than 10 years. Open space was unimproved space transferred to the government. Park was maintained for permanent recreation. Institutions were places of worship, schools, medical facilities, or government space. Commercial was retail, service, or parking lots, and residential was single or multifamily homes. And so we arranged these along a gradient of human investment, increasing from low investment of vacant lots and open space up to very high investment of commercial and residential. So the data, we had 209 400-meter square plots in Baltimore City and County in the US. And this is northeast of Washington, DC. I'm sorry, I don't have a map for you, but I think you probably know where Washington, DC is. It's a very popular location for good and bad reasons right now. So it's in the mid-Atlantic. The total species richness in the regional pool is 116. For the functional trait data, here are our traits. They were taken from a number of different databases and from the literature. They were related to well, various establishment traits and um, life history traits. We used abundance to weight all analyses. For taxonomic diversity. For alpha diversity, we use Shannon index, the Shannon index, and Bray Curtis for beta diversity. For functional diversity, we range standardized all values. I use functional dispersion for alpha diversity and the Gower index or Gower distance for beta diversity. And so we first want to show you the number of unique species by land use. So first up top is total abundance per plot. And I've arranged our plots, and you always see these arranged the same way. Vacant lot, so going from low investment to high investment. So vacant lot, open space, parks, institutional, commercial, and residential. And so abundance goes down from individual trees where there were more influence. When it comes to the number of unique species, there was a lot of variability in composition from one location to the next. People seem to really have a lot of different ideas on what it is that they would like to plant. For taxonomic diversity, alpha diversity was quite high where there was low investment, and quite low where there was high investment. But we saw the general trend that we wanted to see going from low investment to high investment when it came to beta diversity. Institutional, commercial, and residential were generally higher than where there was low investment in or hypothesized low investment in species composition. The pattern became a little weaker. The trend was there, but it came a little bit weaker when it came to functional diversity, and this might be expected because the urban environment imposes such harsh environmental constraints, and so the same traits might be evident even though species composition might be the same. And so functional diversity was the same, not statistically different across all land uses. When it came to alpha diversity, we did get higher commercial and residential turnover. Or, I'm sorry, higher turnover in commercial and residential, but there was quite a bit of variability, and so that didn't sort itself out statistically. So what did we conclude? That beta diversity is higher in residential areas, and we got a lot of number of ornamental species, which was consistent with our predictions. Further, that the multi-scale approach, and this is key, the beta diversity is where biodiversity is being maintained, and this is relative to human influence. And I would contend that composition is more important than richness per se. And so when it comes to sustainability action plans, and when you read them, they talk about biodiversity, and often they focus on, local diversity or alpha diversity. I think I just showed that you can get the same number of species occurring across land use types, and that to me is not as informative as what you're getting in terms of variability, because you get from one location to the next, because you have such a heterogeneous landscape in urban places. And so if you were to return to this conceptual model, and this is published, I think that it is a place to start. I wouldn't say it's the end all, but it's a place to start in terms of thinking about how humans influence the key core ideas that ecologists lean on when it comes to organizing biodiversity, from alpha to gamma via beta. All right, so let me move to a different system, completely different, and come at it from a different direction. So this is going to talk about not how biodiversity can be high in some locations in urban places, but how biodiversity can be low in some locations. And this is from an experimental point of view. So general ecological theory tells us that local communities assemble along an environmental gradient from the regional species pool of colonists. And dispersal is hypothesized to reduce divergence in composition, okay? But there can be rescue effects and there can be stochastic colonization. So dispersal is an important agent to consider when trying to understand community assembly. When you're trying to predict alpha diversity and when you're trying to understand beta diversity. All of this, though, assumes that the regional species pool is comprised of a taxonomically rich community that can exploit the environmental gradient in question. This is a problem.
Chris, sorry, can you just briefly define what is rescue effect, or are you going to do it?
Oh, a rescue
sorry,
effect.
my ignorance.
Oh, no, that's fine. Rescue effect is, so you could have a location. This is actually very important. You can have a location where a species has a population has a negative population growth rate, yet it exists because it is being supplemented by dispersal from another location. So if number of individuals at a location is a function of births, deaths, immigration, emigration, deaths might exceed births, but it's being rescued by higher immigration. That's a rescue effect. Is that clear? Does that help?
Fine. Yep.
Yeah. I didn't hear. I'm sorry.
Yeah.
Yeah, it's great.
Fine.
Okay. Good. So I asked you to return to this figure where on the left-hand side, I ask you to recall this figure. On the left-hand side is this, figure of my cartoon of community assembly. But on the right-hand side, this is what happens when you deplete the regional species pool, okay? And what happens is that you lose the capacity to exploit an environmental gradient. This is not, and this is not, terribly, hard to understand, but if you went to a place where you wanted to create more habitat, okay, but regionally you don't have the species to colonize it, well, then you would never be able to restore those species that you need, because regionally you don't have them in place. And so what I'm going to show you is a test of that by going to a landscape where regionally we hypothesized that the regional species pool had been depleted, creating the opportunity for release of that environmental gradient and seeing whether or not those species can persist, or you get assembly or restoration of those species. And actually, are they there? But just, are they sleeping? Are they in such low abundance that they couldn't persist somehow? And the goal being that, well, if you spend money trying to go to an area where you want to restore habitat and attract species, you're building an area and the organisms don't show up. Is it because you failed in your restoration effort? And the answer is no. It could be simply because the species aren't there to colonize. So urban ecosystems, have these problems. They homogenize the regional species pool. You lose species, you lose habitat. You get this creation of novel habitat, and of course, you get the introduction of non-native and invasive species. So you've got all of these sort of urban syndromes that go along with this problem, that that can deplete the regional species pool. So what we predicted is that low regional diversity and regional scale homogenization of habitat could constrain the potential for local effects to develop interhabitat divergence in assemblage structure. And I'm going to show you how this works. So our questions were, does regional species richness and composition differ between a rural and urban community? Okay, so we compared non-urban or rural and urban zooplankton communities and ponds, and I'll show you those in a minute. And then, does dispersal increase species richness and increase divergence or compositional divergence more along a local environmental gradient for the rural regional species pool than in an urban species pool? And so the study system are ponds, okay? In the Mid-Atlantic region, if you take a look from the air, anywhere we build a development, okay, we build a pond. And these are designed to capture stormwater to alleviate the energy, or to take the energy off of the hydrograph before the water is released into adjacent streams. And they are around roads. So these blue areas, I don't know if you can see my pointer, but they are around roads. They are around industrial areas, and they are around residential areas. And they are all over. There's one right here. There's one over here. They are all over the place. In fact, they are estimated to actually exceed the historical wetland space in the region. And they are habitat. This is the one on our campus. This is our library on the right-hand side. This one is designed to capture rainwater, but it's also an aesthetic feature. And we find all kinds of wildlife in there. Some of them are hidden, and some of them are actually features, aesthetic features. But of course, we have also natural ponds. This one is secluded, and we get quite a diverse, well, relatively diverse zooplankton community. And zooplankton have been studied forever as a great study system, for understanding community testing ecological theory. So we used we developed a pretty substantial experiment using 600-liter tanks. Here they are. This was a 3-way factorial repeated measure study where we had a species pool where we collected individuals from either a bunch of rural ponds or a bunch of urban ponds, where we had weekly additions of dispersers from either rural ponds or urban ponds, 2% by volume. And then we had an environmental gradient. So we set up a stressor. The stressor here was road de-icer. And in the mid-Atlantic US, we get a lot of ice in the wintertime. And this, to alleviate that ice on the roads, we add sodium chloride. And we add a lot of it. This runs off into these ponds, and that sticks around. The salt levels can be actually quite high. The conductivity levels can be quite high. And we know that those conductivity levels can drive away a lot of the zooplankton species, leaving mostly rotifers. It gets rid of a lot of the ostracods, for example. And so we know from a lot of the studies that that we've published, we've looked at amphibians, that we get compositional shifts due to this thing, due to this this environmental constraint. So we ran the study for 12 weeks. We sampled zooplankton bi-weekly. The dispersal events were weekly. We measured conductivity, temperature, water chemistry, and phytoplankton biomass. We monitored all of that to make sure everything was going okay. and I'll show you the uh, the relevant dependent variables as we go on. So the regional species pool, the data treatment we looked at the regional species pool as a two-way repeated measures. We looked at rural versus urban, and I'm going to show you the results for weeks 2 to 12. For the mesocosm study, it was a three-way factorial, rural versus urban, dispersal versus not, and then the road de-icer. Now, the results I'm going to show you is going to be restricted to comparisons on week 12, so just the end of the study within source by dispersal. Okay, this is just to make things easier. So just what happened at the end. So what was the assemblage effect at the end? In terms of composition for the regional species, in terms of composition, so let me go back. That was for richness. For composition, we used non-metric multidimensional scaling for the regional species pool. And for the mesocosm study, we looked at divergence the same way in plus or minus the de-icer comparisons of week 12 within source by dispersal. So this is the regional species pool, just to show you that in terms of composition, this is an NMDS. You're looking at each point represents composition for a dispersal event. If the closer two points are together, the more similar they are in composition. The dark points are the rural sources, and the open points are the urban sources. This is the centroid, or the average of the open points for urban, and this is the average for the dark points for rural. And so this is to show you the weight of the data, or the weight of the distribution of the points for each one. And then this is the convex hull to show you basically the limited space for each one. And so what I'm trying to show you here is we get a good separation in composition between the urban and rural. So we do get differences in species in taxonomic composition between the two source pools. This is the source by dispersal by local effect on richness. So you're looking at, so let me just show you, you've got richness versus time. Okay. You've got rural on the left, urban on the right, no dispersal up top, dispersal on the bottom. And then the two lines are no salt. So I've got salt, which is NaCl, is the open point. And then salt is the dark point. I'd like to point out a couple of things. The first is that when you have no dispersal, the lines flatten out at a lower richness than when you have dispersal. Okay, that's what you can expect from this basic theory. The more you add colonizers, the higher dispersal. The second thing is that where you have rural colonizers, you get higher richness. Where you have dispersal, then you get urban. The main result here is looking at the end of the assembly, where you get separation. It's most pronounced here, where you get with no salt, you get higher richness with dispersal than with salt, with dispersal. So the assemblage trajectory is as predicted, and it's quite strong with dispersal. It's not as strong without dispersal. Going over to the urban side, you don't get that effect at all. Looking at the right-hand side, this is what happens on the last day. Left is rural, right is urban. This is the source pool. This is richness. This is the effect of salt. On the left-hand side, what you're looking at is no salt versus salt in the rural on richness, but you don't see the effect. You don't see any effect of recovery in the urban treatment. The suggestion here is that the urban species pool cannot take advantage at all of the relief of that environmental gradient, or the no salt environment, because the taxa aren't there in the regional pool in order to colonize it. The compositional effects bear out. They're not as clear. I want to show you just two things. On the left-hand side is so the NMDS, this is composition. On the left is rural, on the right is urban source pools. This is for the last day. You've got the dark centroid is salt, the light centroid is no salt. On the left, you see some separation. What you should see is a tighter NMDS for salt than you do for no salt, and that's because beta diversity is lower. The environmental constraint should tighten up beta diversity some. You should see less variability or less compositional turnover going from one pond to the next. On the right-hand side, what you see is basically complete overlap, lower variability, basically no diversions whatsoever as hypothesized. So the results here are the regional species pool for richness. The rural regional species pool was greater than the urban species pool. And for the mesocosm study, the question is, did local factors drive divergence in community structure? And so if we were to organize the results like this, rural versus urban for richness, the question is, yes, it did with dispersal. For urban is no. For composition, the answer is yes with dispersal. What is no? So dispersal was required to overcome the environmental gradient. The effect was only there when you had rural dispersers. The urban regional species pool could not overcome it. So the answer to both questions was yes. Does regional species richness and composition differ? And does dispersal increase richness and increase divergence more along an environmental gradient from a rural pool than an urban pool? And the answer was yes. So what are the implications for both pieces of this work? One is the field of dreams hypothesis, this idea that if you build a habitat, they will come. If you build new environments in degraded regions, but desirable communities may not assemble because they simply aren't there. And so when you spend the money and find out that they fail, it could be that the animals just aren't there to colonize. It may not be that you didn't do a poor job. We've published this in an ecological applications paper when it comes to stream ecology. And I can talk to you about that afterwards if you'd like, in the context of river networks in isolation. The capacity of the regional species pool to be supported on a large scale may be a function of landscape level heterogeneity, and how sustainability action plans invest in biodiversity and restoration may need to be a multi-scale effort. So you have to think not just locally, but regionally about the context where you put things. And so to conclude, beta diversity, I think, is key to understanding patchiness and human influence. It links diversity patterns across scales, and it suggests why diversity is high in cities for some taxonomic groups and low for others. And I think it has implications for sustainability policy because of this plot right here on the right-hand side. Because if you understand dispersal, you're connecting regional diversity to local diversity, because you're understanding compositional shifts, because the agents of change are important to understand because they target what sustainability action plans need to target. So with that, I want to thank you very much for listening. I hope I was clear. I hope you understand my work and one aspect of biodiversity that I think is important. I'm happy to answer any questions. Of course, take any kind of critical questions that you might have. There are some folks here that I need to acknowledge. I need to acknowledge the National Science Foundation for funding some of this work, and of course my institution. And there's my contact information and website if you want to look at things some more. Thanks so much, everyone, for sticking around late in the afternoon. I think it is for you. It's still early here for me.
Well, thank you, Chris. I mean, it's still a working time. It's not like holidays. And well, it's I think it's you use quite advanced community ecology theories, and I don't know whether everyone was up to date on this. But I mean, discussion is not only to show. I mean, it's also to show that something was not clear, and maybe Sven, Chris can go back. And actually, I have a question which I didn't get totally. So in the experiment, in the pond experiment, you mentioned dispersal. So was it like just taking water and adding waters, but you might also add species that they usually wouldn't, or they are low dispersal. And so usually dispersal is like something active, and if you are not able to disperse, I mean, you stay where you are. But then by adding, you add both good, well disperser or good disperser and low disperser. Does it make some differences in, or since you standardize this everywhere, then it should be no problem?
Yeah, so there are these assumptions that you make about dispersal, and the assumption is with zooplankton, and so this has been done in other experiments, and there is no standard way to do it. Mm-hmm. There's a couple of different studies, and we went on the safe side in terms of the amount. The assumption is that all zooplankton will get picked up and could potentially get picked up and moved by a bird or the wind, and that there is no bias. No one knows. No one knows. Mm-hmm. And so I think your point is well taken. No one knows, and so what we did was just did it equally and see. I think that if our results did not bear out, then we would have to explore that more. But since our results bore out, I'm less concerned.
Mm-hmm. No, I mean,
since it's, but that doesn't mean it's real. You know, that doesn't mean it's real. It just means it's experimentally real. I mean, yeah, I mean, one way to do it is you can take buckets and you can set them at different distances from a real pond and fill them with water and see, you know, how they blow, you know, how they collect naturally. Or you could collect birds and, you know, wash them off and see what kind of, and then compare them to what's actually in the water. But yeah. No one knows. No one knows.
I mean, other want to raise questions?
Please, if there's any questions, you can raise your hand.
Okay, yes. There's this
also virtually.
Or also write in the chat.
Or also write in the chat, yes.
Mm.
Kevin?
Yes, you might.
Can you hear me?
Yep.
Cool. Hi, Chris, really nice talk. I found it really, really fascinating. I really liked I like to see stuff also in the experiment. It's something I always wish I was able to do a bit more. I also work on sort of urban, or I did for my PhD, urban biodiversity, but on wildflowers. So like spontaneous vegetation as well as sort of just herbaceous stuff in Zurich. And I found very similar stuff in general. The importance really of looking at beta diversity and not just alpha diversity, because that's where I really found like these big differences. I was mostly focused on size, but it plays into the same sort of categories, because a lot of the small stuff are maintained by the city and thus have really high beta diversity, much higher than you would expect for their size. And talking about dispersal, I tried to look at some of this. I just didn't have the data because I was very interested in choosing species that can disperse on their own and what effect that has on sort of spreading biodiversity in terms of like an intervention by management. And I think your work highlighting the role of dispersal is quite interesting. What are your thoughts then on sort of this aspect of it? Because I mean, in these residential areas, in these areas where there's management, there's species selection and sowing and stuff like that. Do you think there is a way in which we can sort of like promote species that are good dispersers, that maybe are native good dispersers that we're trying to see more of in our cities rather than other types?
I would answer it two ways. The first is, I think we need to learn whether or not the natives can exist in places that we want them to exist in.
Sure.
So we did some work in vacant land. I don't know what the situation is with vacant land in your area, but in Baltimore, we have a terrible problem with it. Where houses once stood, then we knock them down, and then we leave these soils that are about, a few centimeters, deep, and then it's just concrete and terrible stuff for meters. And we found that, some of our native plants could exist there just fine. The other is, and we've got a paper, I can't remember if it's published or if it's in review right now. It's one of my students' dissertation chapters where we describe the dispersal syndromes of a whole slew of urban species. And so the second part of my answer would be like, we just need to know what they are. Like what are the typical dispersal patterns that we see. And a lot of it was like ant dispersers, wind dispersers. We found a lot of animal or dispersers, and that was because, and it's not animals, it's humans, like getting caught on like human, like pant legs, your legs as you walk through. And so I think that kind of like basic ecology just needs to be described first, and then that information needs to bubble up to the top into these folks that do the management. I guess that's how I would. It's probably not what you were expecting me to say, but I think that those two pieces of information need to be studied and then delivered to the policymakers first before they can start doing what I think you probably wanted me to say. I don't know.
I mean, no, I think you're 100% right. Sorry, I just like we don't know enough, and we don't. It's, even for stuff we can say it's wind dispersed, but how does that look in a city that's like New York?
Right.
And so like with these columns, these canyons, and so absolutely there needs to be a lot of this basic research done. Still, I think so. Yeah, no, I think it's a great answer.
Other questions from the audience? I was wondering about regional species pool. I mean, you found a way to define for plankton, but what about plants in cities? So how would you consider a species pool? It's like the whole world, because I mean you can get plants from everywhere, from the internet, from garden centers, nurseries. So how would you consider this?
So, for community ecologists, that think about local regional effects, it is the elephant in the room, the regional species pool. You know, we start talking, and if someone brings it up, and it's just like everybody turns the other way and just doesn't look at it. The regional species pool, you know, we define it as the list of species we have in our data set. But there is the species that are not there, that could be there, that you're not seeing, that also matter. And because if a species is not there and you're not seeing it, that also tells you about environmental filtering. It tells you what species could not exist. And therefore, even though you're missing it, it doesn't mean that, it means that there could be a species that there could be a niche out there that you're not seeing, that you're not observing. I think they call that dark diversity, I think. And so no one, there's no way to know. There's just no way to know. Some people, like when it comes to urban places, will go to the garden center and just start writing down all the species. Or they'll pick up a catalog of garden plants and just start writing them all down. I've thought of like fantastic, I mean, there's the great studies where you could do like look at garden catalogs through time for a location, and look at the evolution of, you know, what has been the change in species composition in the designed regional species pool for an area, and what does that tell us about the ecology of a region? And you can do functional traits, you can do, you know, phylogenetic, you know, composition, you can do all kinds of things. There's so much armchair science you can do, but I mean, I'm going on a tangent, but there's no way, there's just simply no way. I don't think there's a way to completely know the regional species pool when it comes to any ecological study, period. You can only get close. You can only get close. And it is an argument, urban, non-urban, it's you can't get at it. You can't get at it. You just can't. I think it becomes harder with urban places.
Yeah, I mean, defining the species pool sometimes, I mean, it's crucial, no, to understand this dark diversity, or to understand why a species is not there. Maybe it's filtered by humans, and I mean, considering humans as a filter, because I mean they select species based on colors, based on
Yep.
Allergenic or whatever. So do you think that because you were talking about mechanisms, and we understand a lot about mechanisms related to dispersal, to ecological niches, and drought resistance, and things like this. But I mean, human is really, I mean, you demonstrated in your diagram this social investment started by selecting plants based on preferences, based on expectation, or maybe norms or education. So do you think we have to revise our concept of traits, since traits were like always like referring to plants for to maximize reproduction, growth, and survival? So do we need like a lot of traits, social relevant traits, or at the end are always the same? I mean, the traits, the definition of traits is independent, whether it's like selected by human or selected by the environment, is always the same traits? Or do we need to consider a bit this concept of traits?
I think we need another axis, which is preference. I really do. I do, and I think that the landscape designers have done it. You know, I think that we can turn to them and come up with another, just list of functional traits that are, I mean, we can just call them something else, preference traits, something like that, design traits, like my wife, probably doesn't care that I've got, 4 different species of evergreen in my front yard. She just cares that they're all green all year, so it doesn't have anything to do with, their life history for her. It's just that they're green. That's just an example of what we're talking about.
No, I was just wondering, because traits is really a super nice tool that maybe concise social ecologists and social scientists and ecologists, and it break barriers between disciplines. But I was wondering whether we have to also to merge other traits, and I have my personal opinion that I mean, for a plant, traits have to have a link to fitness. Then, if it is relevant to human, colors maybe is not so relevant, except maybe for herbivores or for frugivores. But I mean, for humans, is very important. Whether this filter based on the color has an effect at the end on the growth and survival. I mean, this might affect other traits that are more linked to the functional, to the real functional traits. But at the end, I mean, maybe as you said, we have to change or to add a new axis, which is the perception or the preferences. But the list of traits, I think, I mean, those that really are relevant for the plants to survive or to perform remain the same or not.
Yeah. And I think that, furthermore, if you do it, I think we can make predictions about beta diversity when it comes to it. Beta, whatever kind of, if you want to call it, functional preference diversity or something. I mean, beta diversity and functional traits for the trees didn't change that much when it came to people, versus where people weren't having a lot of investment. And that makes sense because, the traits should have to be the same in an urban environment because of the stress. When it comes to preference traits, you might see a lot of functional diversity, functional turnover, which would be, consistent with social theory, probably.
Mm-hmm.
But if you looked along socioeconomic gradient, you might see something different, and that might help us understand, something else about biodiversity. I think these are important questions to ask, I think.
Mm-hmm.
And not all that hard. I don't think that hard to do. I mean, you can put an undergrad on the cataloging of the traits and do the analysis quite efficiently, I think, and have an undergrad involved to do it, I think.
Okay, thanks, Valentina.
Yeah.
What do you think? Yeah.
I think our time is up. But thank you so much, Chris, for a super cool and extensive presentation. This was amazing. Yeah, this will be it for us. You can stay with Marco for a little longer.
I mean, everyone who wants to stay, yeah.
For further discussion, but I officially close the recording.